Friends, welcome to Biology Made Easy. Today we will discuss about transcription. What is transcription? It means the formation of RNA from DNA. But now the question comes, why this is necessary? We know that the DNA contains all the genes, and the genes are responsible for all activities of the cell, and all the activities of the cell are regulated by enzymes, and the enzymes are proteins. So these proteins and enzymes are synthesized by the DNA, where the blueprint is there. That means the DNA behaves like an architectural engineer. It has the blueprint, what type of proteins and enzymes will be synthesized. But the DNA is always present in nucleus. It never comes to the cytoplasm, to the ribosome, where the proteins will be synthesized. Then how the whole thing will take place? That is why the DNA, which behaves like an architectural engineer, it sends one messenger, which is known as messenger RNA. It behaves like a contractor, and the mRNA will carry the message from the DNA, and then that will be translated into the language of proteins in the ribosome. This is known as transcription. That means, suppose this is the DNA, then this DNA will transcribe mRNA, and then this mRNA will transcribe into proteins, polymer of amino acids. So this step is known as transcription. Now, how this transcription takes place? What is the principle? Just like replication. In a replication, what happens? Suppose this is 3' to 5', and this is 5' to 3', two strands are there, and both of them are arranged according to their complementary base sequence. The same thing happens during transcription. When one of the strands will be transcribed, suppose the 3' to 5' will be transcribed, then the mRNA which will be synthesized, that will have complementary base sequence. Here it is 3', it will be 5', 5', this will be 3'. And suppose here the sequence is G, C, T, A, like that. Here G is there means here it will be C. Here C is there, that means here it will be G. T is there, here it will be A. And here A is there, so since it is mRNA, it will be U. So same complementary base sequence. But again it differs in some respects with the DNA replication. In case of replication, both the strands will be replicated. But in case of transcription, only one of the strands will be transcribed, not the other one. I will tell you the reason. The second point, in case of transcription, the entire DNA is replicated. But in case of transcription, only some segments of the DNA will be transcribed, not the entire one. Why? This is the DNA. In this DNA, several genes are there. Here one gene is there, here one gene. Likewise, so many genes are there depending upon the number of chromosomes and the type of chromosome. So, suppose you take the case of Y chromosome. Y chromosome has 231 genes. Take the case of chromosome number 1 in human being, it has 2968 genes. So, do you think that all these genes present in this chromosome will be transcribed at the same time and will be translated to different types of proteins? No, they are not necessary. That means the protein which is necessary, that type of mRNA will be transcribed. Suppose this part will be transcribed, and here another will be transcribed, the remaining will not be transcribed. That means instead of both the strands, only one strand will be transcribed, and again only part of it will be transcribed, not the entire one. So these are the two differences between replication and transcription, but the similarities based on the principle of a complementarity. Now the next question is, if both strands will transcribe, what will happen? Why both strands do not transcribe? Now see, I am drawing here one DNA. This is 3 prime to 5 prime. This is 5 prime to 3 prime. Suppose this is the strand which will be transcribed, and this is known as template strand, or it is known as non-coding strand, or it is known as nonsense strand. While the strand which will not be transcribed, this is known as coding strand or sense strand. So suppose here it is T, then here it is A, here it is C, again G, again C, then here it is T, that means here it will be A, here it will be T, C, G, G, C, then C, G, like that. Both are complemented to each other. Now, if both strands will be transcribed, then this side will give rise to an mRNA. Here T is there, that means here there will be A. A is there, here it will be U, because mRNA. C, then G, G, C, C, G. And again, in this side, if it will be transcribed, suppose in this side also it will be transcribed. Here A is there, U, T, A, G, C, C, G, G, C. Now you see what is happening. This mRNA is complemented to this DNA strand. This is complemented to this one. If this mRNA will translate into protein, then the codons are A, U, G, C, G, and so on. So it will give rise to one type of protein. The other one is U, S, C, G, C, and so on. It will give rise to a different type of protein. That means the same gene will give rise to different types of proteins, which is against the genetic law. That is one of the reasons. Now you come to the second reason. This DNA strand is complementary to this. Now this template strand or non-coding or nonsense strand is complemented to this mRNA. And this strand is complemented to this one. That means what is the relation between these two mRNAs? They are again complementary to each other. These will be complementary. Here you see there is A, and in that side it is U. In this side it is U, that side A, here G, here C. That means AU, UA, GC, they are again complementary to each other. That means it will give rise to a double-stranded mRNA. And when a double-stranded mRNA is synthesized, it cannot be translated into the language of proteins. So there will be no function. So these are the two reasons why only one strand is transcribed and only segment of it transcribed, not both. Right? Now, when one of the strands will be transcribed, this is the 3' to 5'. This will be transcribed known as template strand, non-coding strand, and the nonsense strand. The other is coding strand. Here different types of questions are usually asked. Suppose, remember, it is only the non-coding or nonsense strand that is transcribed, not the other one. Sometimes if in the question will be asked, suppose the sequence of the coding strand is given, then what will be the sequence of mRNA? You mark it. This is 3' to 5', means this is 5' to 3', this mRNA. But this side you see, this side is 5' to 3', but what will be about this one? This will be 3' to 5'. Now you see the relation between the coding strand and the mRNA. Here A, T, U, because in RNA T is not there. So A, T, U, G, G, C, C, G, G, it's same. That means the mRNA which is transcribed from the non-coding strand is exactly similar to the sequences of coding strand except T is replaced by E. This question is very important. Now, the next thing is, when a transcription takes place, which enzyme is responsible for it? The transcription takes place due to RNA polymerase, which is DNA dependent RNA polymerase. Now, that polymerase, when it will transcribe, during this transcription, there will be three factors. One is promoter, the second is structural gene, and the third one is terminator. If this part of the DNA will be transcribed, then this is the starting point, this is the terminal point, and this is the structural gene. So, in the beginning, there will be one promoter site, and in the end, there will be one terminator site. But in a DNA, how they are arranged, that will be more clear when I will draw a second diagram and I will explain it. Now you see, these are the two strands of the DNA. This is one structural gene, and this is the template. Then you see, this will be transcribed to mRNA. The mRNA will be transcribed in this manner. Now you see, since this is the structural gene template, that means this is the coding strand. Now, on either side of this structural gene, there are two things. One is promoter, the other is terminator. Now, which one is promoter and which one is terminator? This is promoter and this is terminator. That means the promoter always lies to the 5' side of the coding strand. That means this strand of the DNA runs from 5' to 3'. That is, 5' is the upstream and 3' is the downstream. Just like you think of a river. From upstream it is moving to the downstream and toward the ocean. So in the same way, this from the 5' it is running to 3'. That means this is the upside, that is downside. That means the promoter always lies in the 5' upstream of the coding strand, while the terminator lies in the 3' downstream of the coding strand. This is very important. Then you may think, if this is the pattern of arrangement of promoter and terminator for this gene, what about the other gene? For the other gene, it may be different. Now, suppose in this case, this strand will act as a template. Then this will be the coding strand. So, if this is the coding strand, then the 5' lies to this side. So naturally, this will be the promoter. This will be the promoter, and this will be the terminator. This will be the terminator. That means you see the position of promoter and terminator can be switched to any side, and depending upon that, which DNA template, which DNA strand will act as a template, which will act as coding strand will be determined. You see, for this gene, this is the coding strand, and this is promoter, this is terminator. But for that gene, this is the template, That means this will be transcribed. This is 3', so it will be transcribed in this way. And that is the coding strand. And again you see the promoter lies in the 5' upstream of that coding strand. That means the position of a promoter and terminator can be switched, and accordingly the which one is coding strand, which is the template can be determined. That means again I want to emphasize that out of the 2 DNA strands, one should not think that only one strand will be always transcribed, the other one not. Not like that. For one gene this may be transcribed, for the other gene the other strand may be transcribed, as I have explained it. Next thing, this transcription, as I already have told you, it is catalyzed by the enzyme RNA polymerase. This RNA polymerase is a DNA dependent RNA polymerase. Now this enzyme is a hollow enzyme having alpha, beta prime in the core part. This is the core enzyme. While there is one sigma factor which is added to it. Suppose this is alpha, these are the beta prime, alpha, beta prime. This is the core part. When the sigma factor will be attached to it, it will be hollow enzyme. Now, what is the function of this alpha, beta prime and sigma factor, and how the initiation, elongation and termination of this transcription take place, we will discuss in the second part of this. This is part 1. In the second part, we will discuss the details. Now, I will give you 2 questions from this part. Now, question number 1, which is very important. If the sequences of coding strand is 5', A, G, C, T, A, G, 3', then the mRNA will be A choice, 3' A, G, C, U, A, G, B choice, 5' A, G, C, U, A, G, then C choice, 3', then U, C, G, A, U, 5', then the next one, no, not the above. This is very important question. Second question, how transcription differs from replication? A choice, both strands transcribed, second, entire genes transcribed, third, segments of DNA transcribed, last you can write none. These 2 are also very important and good questions. I hope you will be able to answer it. Thank you very much for watching it. Have a good day.